So during the next half an hour or so, the idea is to first give a very short presentation on the key features that we have developed within Phenostore, this phenotypic data module. And then I will be giving also a short demo on how to enter a case in the system. And after that, together with Stephen Lowry, that he's here with me, we'll be answering questions, and we are also open, as Sergio was mentioned, to discuss on other features that you think could be interesting to have in the system and that might be missing at this stage. So Phenostore is meant to be a user-friendly tool that will enable you to collect, share, and also export standardized phenotypic information, and it has been built mainly for rare disease patients. And now I will be mentioning and giving some example of the different features that the system enables. So first of all, the system will gather the information, the clinical information, using standards and ontologies. And for that, mainly, we use the Human Phenotype Ontology, the HPO for the symptoms, and also the Orphanet Rare Disease Ontology for the clinical diagnosis, and the OMIM database for the molecular diagnosis. But apart from that, the system can be customizable in some ways, so that relevant information from different types of disorders regarding, for example, biomarkers or type of tumors, as we'll see an example later, can also be entered in the database using structured vocabularies. So this can enrich the data you have for your patient, and then the use that you will be giving to it. Another important thing on how we have built the system, and Sergio has mentioned, is the interoperability. Not only by using the standards and ontologies, but also because we provide APIs, so programmatic access to the system. And this helps us to fully integrate this clinical information and this phenotypic data with the GPAP and with the genomic analysis. But this will also enable you to use it for other type of omics analysis. The system also will enable you to have an overview of all your cases, to browse your data, and to, let's say, play a little bit with all the information you have for creating cohort and other type of possible new analysis, starting from the phenotypic information. You will see that the system enabled to bulk upload and import information, meaning that if you already have your data standardized in your own database, we offer means to quite straightforwardly import this information in this system, so that you don't have to re-enter everything again. And more importantly, the information that will be inside Phenostore is not just closed in the system, but we're able to export this data using PhenoPackage, which is now the standard for phenotypic data export. And this will really help you to have your data standardized and to be able to reuse it also in other systems that accept PhenoPackage and the standard for phenotypic data export. So to facilitate the phenotypic data entry in the system, we propose 20 different disease forms that are based on the Genomics England data model, and you will see later what does that mean and which parts are tailored to a different type of disorders. But the idea is that all the rare diseases should be represented there, and also in collaboration with the Orphanet team. And within the SolveRD project, what we have done is to map the different European reference network to these different types of disorders, so that can help clinicians to decide in which study their patient will belong. So as we were mentioning, in these different forms, you will have options to enter phenotypic information of your patient, and this is tailored to the disease types. You will see later in the demo this in a more detailed way, let's say. But the idea, for example, is that you will have a list of HPOs that match this type of disorders, and this will help you to do deep phenotyping of your patients, but also to manage differential diagnosis, because some of the signs and symptoms might not be present in your patient, but you know that they might be associated to that disorder. And as we mentioned before, and the example we are showing here is regarding ERNS and Tourism and a collaboration with it within SolveRD, as they are interested in also gathering information about the type of polyps and the number of polyps that the different patients might have, and also some specific tumor features. So we have enabled the possibility for them to enter this information in a structured way that they will be able to use in the future to structure the patient and create a specific cohort based on this important phenotypic information. So to import the data in the system, there is a workflow that consists in 4 different steps. We'll go into more details later, but basically, first you will be choosing the template, so the disease type your patient, your case belong. Then you will be entering the clinical information of your index case. We'll draw the family pedigree and associate to the members of the pedigree other relevant clinical information. And these steps can be done within the user interface using a case-by-case approach, and this is relevant when you have a few cases to enter or you don't have your data already standardized, and we'll see an example on how to do that later. But as we mentioned at the beginning, we also enable to bulk import your phenotypic information. So if you already have this data in a standardized format in another system, we'll provide different means to directly import it through Excel files. So we provide a template where you will be able to add all this information, but we are also compatible with phenotypes and the JSON files that they export. And we are also currently working on being able to import these pheno packets directly on our system. Once you have entered your cases and your information, you will have summaries and the pedigrees we were mentioning, and you will be able to export also the information from the system, either as a PDF to keep a record on your site, but also, and more importantly, using a pheno packet so that you have your standardized data interoperable with the other systems. And the system already enables, of course, to share your patients and your data with other RD-Connect members. You will also be able to have an overview of your cases, filter and export your data. And as we were mentioning, we have just last week released this phenotypic module, Phenostore, within RD-Connect. So we have now full integration with the RD-Connect genome phenome analysis platform, and this will enable to use this phenotypic information to filter genomic data and also to prioritize variant. And we are also linked to data discovery networks such as Matchmaker Exchange, and this phenotypic data will also be used for that. Another tool that if we have time, I can very quickly show you after also. We have a beta version in production, but we are meant to release a new full version in the next weeks. Is a cohort application. So all the data you will be entering in Phenostore, that you will have access of your own samples. You will also be able to create a specific patient cohort within the whole RD-Connect database, and this based on your samples or samples from a specific ERN or a specific project. You will be able to filter the data according to any type of phenotypic information, and then create your cohort based on that. And also according to the experiment information, for example, if you only want to analyze exomes, genomes, or specific kits. So with this application, you will really be able to create specific cohorts for doing downstream later analysis. And finally, before doing the real demo, just as Sergi was mentioning, to thank really Davide Pisce, Alberto Corbo, and Anastasios Papakonstantino, which are the ones that have developed this tool. Okay, so now I will stop sharing 1 second. And okay, can you see my screen again?
Yes.
Okay, perfect. Okay, so now the idea would be to show you how to enter a case in the system and also a couple of other features, and then we'll have time for a discussion with Steve. So this is the landing page of the RD-Connect platform. Just to tell you that now here on the top you have like some kind of summary of this release, and you have access to some preview videos and a couple of tutorials that we have already made that should help you to create an index case or edit the pedigree, but we are meant to do some other videos like that with other features. And to access the Phenotypic module, it's here at the middle of the page, and you also have a link to the submission guidelines with really detailed explanation of each of the steps. Today we'll be using for the demo the playground version. You can also use it if you want to have a look at the system and you are not an RD-Connect user. Okay, so this is the main page of Phenostore. So as I mentioned before, we have mainly two options to import cases in the system, which is the bulk upload of the single case entry. For the bulk upload, I will just quickly go through it, but you will just need, okay, I have to log in. You would just need, if you want to enter the information using an Excel file, to download the template that we provide. And then you will have to fill in the same information indeed that you will be seeing now once we enter a single case. I just want to show you here that we provide also drop downs to ease the entry of this information, and the red fields are the ones that are mandatory on our side to upload the data. But once you have filled in this template, you just need to upload this file in the system, and we will be automatically creating all these entries in the database. But for the purpose of this demo, and also for you to see which are the fields that we are currently collecting, we'll be entering a case, an individual case through the user interface. So you will be clicking here on create a new index case. Here you have a summary of the first step that we were mentioning. So the template, entering the phenotypic information of the index case, draw the pedigree, and add family member information. Here is the list of the 20 Genomics England-based templates that we are providing. In case you wouldn't find any of disorder that really fits your patient, you are always able to select the default template which has not been tailored by any type of disorder. For the demo, I will be entering a case using the neurology and neuromental disorders template. So once you start creating an index case, we always start to enter the information through the index case. Then you have a 8-step process with we will be asking different types of information. Only the individual information from this first page has mandatory field, and the other one is more related to additional clinical information that will be useful for downstream analysis too. So now I will enter the patient information. You can also enter the country of birth. The year of birth is mandatory for index cases, but not for relatives. Then the sex and the affected status. When an individual is affected, we then ask to answer if the case is solved or not, so that we also keep track of this in the system. To go to the different options, you can use the button here next at the bottom of the page, or you can also directly navigate through the workflow at the top. The next step would be the patient consent, and here it relates if you want this patient to be discoverable through matchmaker exchange or not, or to prevent communication on incidental findings in the genomic platform. Then you can enter family history, And here you will see that the different information, for example, for the suspected mode of inheritance are HPO encoded. So we are using already the ontologies to be able to then reuse this information. And you can enter as many suspected inheritances as you want. We also have, we also collect information, for example, about consanguinity. And if a proband is suspected to be consanguineous, you can also enter the degree of consanguinity of the parents. And then you can enter other type of family information. Concerning the medical history, the same as before, we collect all this information using HPO, so the age of onset, and also the progression of the disorder. You can also upload to the system any other additional documentation that can be useful for the clinical diagnosis or to have on the system, but please remember that these documents cannot contain patient data or metadata that can identify the patient. Then you can also enter measurements, and you can enter as many entries as you would like. So once you select the day when the measurement was done, you can then, once you add the information about the height, the weight, and the head circumference, there are automatic charts that will be appearing. So that you can already have an overview of your data. Here, the next field is on the signs and symptoms, and this is where most of the form are tailored to the subtypes of disorders. So here we have selected the neurology and neuromental disorders form, and we have several subtypes of these disorders. So if we have a case that has a neuromuscular disorder, we would select this field, and then the HPO associated to this type of disorders will appear, and you can browse the HPO ontology to do deep phenotyping and add all the relevant information from your patient. Once you select an HPO, this one will appear on the right panel, and you will be able to select if this symptoms or sign is observed or not observed. And when selecting observe, you will also be able to add the severity of that symptom. And this is also HPO encoded and can also be exported in an interoperable way and reusing for other analysis. If you select HPOs that are not observed but are relevant for differential diagnosis, it is also important to enter in the system. But also, if you don't find in the list your HPO of interest, you can always, and in all the form, browse at the top the HPO ontology and add the corresponding symptom. You can add as many symptoms as you like, but generally we require 5 or 6 HPOs in most of the type of disorders to be able to give a good phenotyping of the patient. So the next field is related to the genetic testing, and we gather previous genetic information so that when you will perform the analysis in the GPAP, you will quickly know if some studies have been done before. For example, I can select the RI-CGH and just comment that the RI was negative. But also, if I have already screened a relevant gene that is normally associated to the type of phenotype of this patient, I can also enter this information and say that the gene has been negative. And for example, I've done a single gene screening. So all this will be available when we are performing other types of analysis, and you are aware of it. And finally, the last field relates to the diagnosis. So to enter the clinical diagnosis, as we were mentioning, we use the Orphanet Ontology, or the ORDO, and you can also here browse the ontology to enter the information. You can add as many ORDOs as you want. Sometimes the diagnosis might not be confirmed, but suspected. And the same with the molecular diagnosis. Normally we use the molecular diagnosis once the causative variant has been identified, but you can also enter several OMIM entries, so with a suspected diagnosis status, so that when you do the analysis, you are aware of what you think could be the origin of the disorder. Also, you have a free text box to always add any type of relevant information that you think could be useful for the analysis or your case. So once you have entered all this information, you can submit your case here using this submit button. And then a family will be created, and you will be able to either add relative if you have more information about the pedigree, go to browse your cases, and we will do that later, or you can directly go to see the summary of your case. So now we are going to add a relative, and this brings us directly to the pedigree of the patient. So we already have the information that has been entered, that is translated in the pedigree. So we had an affected female. At the top of the pedigree application, you had here the instruction on how to enter any other type of information, parents, children, siblings, partners, but also I will show you how to identify consanguineous relationships. So now just to quickly show it, I will be adding a sibling, so I will add a brother. If I want to include more information about this brother, I can directly select here in the settings. I click, and I can either link it to a patient that is already in the database, or I can add more information about that participant directly here from the pedigree. So here I can say it's a brother. I can enter also the date of birth and the affected or unaffected status. Once I update the case, I can just keep the information in the pedigree without creating a new form. Or if I want to create a form, either because I'm also submitting genomic data or because I want to add more detailed phenotypic information, I just need to click here, yes. And then a Phenostore form is directly created, and I could, from the pedigree, directly jump to it and add more information if necessary. Just to show you from here, you can also identify a consanguineous relationship by dragging this line, and this automatically creates children. But if I want to delete any entry, I just need here on the top right to click and delete that individual. So once I have the pedigree, I can always come back to my index case by clicking there. I can export this pedigree in JSON or PET formats, and I can also manage the permission of the case to share it with other collaborators within RD-Connect. If I just quickly go back to my case, I just want to show you that, as mentioned, you can export all this information using the JSON format, but or also the Phenopackage standard, and I can save it and already reuse it. And I can also have access to this case summary, where you will be able to export the PDF with all the information that has been entered in the system and keep track of it. Okay, so once you enter the info, also I would like to show you that you are able to browse all the data and information that you have entered in the system. So here you have a table with all your cases. You can see as many cases as you would like to. On the top of the screen, you have some graphs that get automatically updated once you filter the data, and you can filter it using these bars in the column. For example, if I want only the information about the index cases that are entered in the systems and the ones that are unsolved, I can quickly filter that. And if I want, I can export this data to keep it, reuse it, or to have also this information in place. And yeah, I think I will leave here the demo for Phenostore. I will just very quickly show you what I was talking about the cohort application, just to show that it's in process and being developed. And the idea is that from this cohort application that will be integrated also in the system, you will be able to filter all the data by project, ERN, but also to clinically explore that data based on the ORDO, HPO, and OMIM entries. Here are, for example, the number of patients that have all of these HPOs, and you will be able to intersect or not this data to create your cohort and export all this information also, or directly send it to the GPAP for genomic analysis. And I think I will keep it here, and then we can take questions and discussion.
Thank you very much, Leslie. So if you have questions, well, Birte, I see she has one question in the chat. It says, when adding HPO terms, two clinicians at the same time get feedback on how good the phenotypic description is. She's referring to the fact
yes.
of the page.
Okay, I will keep sharing my screen. I don't know why I stopped, sorry. Okay, so yes, when you enter, now I'm in the playground instance and it's currently not working, but I will show you how this works. So Birte was referring when you are entering HPO information, here we have a link to the Monarch initiative that gives you an indication with this 5-star rating system on how good is the phenotype. Good meaning that how specific it is compared to broad description that might not be very specific and that might be more difficult than to use for downstream analysis and to arrive to a shorter list of genes to assess, for example.
Okay, thanks. Thanks, Birte. We have another question from Estrella. Will it be possible in the future to push data from Phenostore to Phenome Central?
We have not directly thought about this, as we use Matchmaker Exchange, and Phenome Central is one of the links we have in Matchmaker Exchange, so our data is discoverable in both sites. So I don't think we will be having this functionality specifically from Phenostore, but you would have the same application using Matchmaker Exchange through the genomic platform.
Okay, thanks. We have a question from Andrea also. How do we get username and password?
For that, you will need to register to the RD-Connect Genome Phenome Analysis platform if you are not registered yet. Otherwise, with your credentials, you are able to access Phenostore, as now we have migrated all the data from one system to another. So anything you have entered before in the Phenotypes instance is now available here in Phenostore. But if you'd like to register, then you will need to access the main platform page, and there you have links to register to the system.
However, if you want to play with Phenostore in the meantime, this what Leslie showed you here, the playground. She put it in very quickly, but I think we have it documented on the page. The username is test, and the password is 1234, and then you can see exactly what Leslie's done today, and repeat the process for yourself.
Yeah, I think if I click here, yeah, here you have information about the username and the test that Steve was mentioning.
And also to register to the platform, know that you have to be the PI or group leader of the group. So if you're, for instance, a PhD and you want to access the platform, you should ask your PI, and he can apply on the homepage in this user registration site. He can apply to register to the GPAP. So we have another question from Jose Garcia. is already available for everybody the option to share a family information to somebody in particular?
Yes. Steve, correct me if I'm wrong, but in principle, so once you should have a case open. Yeah, now it's not a case. I will pick a case so that you have the information here. In principle, with these permissions, you should be able to share their data to any RD-Connect group. I don't know if I'm answering the question or it was another type of sharing.
The only thing, instead you were referring to a person, you cannot sharing to an individual person. You can share it to a group. So I don't know if you were referring to a particular individual in the platform.
So this is one thing that's changed in our permission system now, right? So within Phenotypic
Yes.
when we were using it, you could choose to share with another Phenotypic user or with a group. But the way we have the RD-Connect GPAP set up in general, all permissions are at group level. So here you would be choosing to share it with another group rather than an individual.
Thank you, Jose Garcia. So is there any other question or thing that you want to know or ask us about Phenostore? You can unmute yourself also if you want to ask something.
Hi, this is Anna. I would like to know, can you also filter in Phenostore for patients that have, for example, in Genturis, the same tumor types? This would be very nice and important in the future for us.
Yeah, you mean if the tumor type has been entered using the GPAP ontology?
No, I mean, we have this polyp information, for example, and there it would be interesting to see if a patient that has adenomatoous polyposis, if he only has polyps of this kind or also polyps of the other kind.
Yeah, sure. It's not possible right now, but we have it in our to-do list pending, because this is really something that we want to enable you to filter by all these customizable fields that you are to make you able to use it for doing this cohort or stratify your patient. So not yet, but yes, you will be able to do it.
Okay, thanks. And another question would be, do we always have to enable other users to see our phenotypic information of the patients that we shared? Or will there also be, for example, in Genturis sharing policy that we can see the patients of the others without always asking them to share it with us?
The idea we have now is to enable this through this cohort application, and this is why I really quickly show it, but I wanted to show this application because it's from there that you will be able to select the ERNS Genturis, for example, and you could retrieve all the information from those patients. All the information that is entered in standard ways, but the information concerning, for example, date of birth or ethnicities won't be available there because it can be identifiable for the patients. So yeah, so this is why we have a separate cohort application for that. But in principle, we think it should enable you to do all the type of analysis you would like. Otherwise, let us know if there is something that you would like to do, but it's not possible there.
Yeah, maybe as an addition to this, I mean, in the system, you have ways of contacting other users and then telling them, you know, I want to know more about this patient, so please share the full phenotypic profile with me, and they will be able to do this now through Phenostore. But also, I think that, I mean, basically we're putting out this concept now and this new tool. But if there are some projects some ERNs that really want to share across the whole set of users all their data, and they have the consent of doing this or the agreement to do this, I think there are ways that we can enable that, right? In a similar way as what we are doing is enabling, for example, the sharing of the genomic data between also of RD partners. So I think that's doable. We just need discussion, and we need instructions from the coordinator of that ERN or the data set of the project to do that, and then we can discuss it and we can build some automatism so that basically what you would do is automatically share all the data sets with all the partners that belong to that project or that ERN or whatever. So that's something that we can discuss.
Okay, thanks. I think what Leslie already said would be sufficient to just share the phenotypic data and not the data like birth date, because this is nothing I'm interested in right now. But yeah, we can talk about what our needs are, and it is great that we can share it in the future. Thanks a lot.
Any other question or comment that you want to address? Okay, so I guess that we can finish this webinar here then. Thank you very much all for joining. I hope it has been useful for all of you. And if you have any further questions or comments, of course, you can contact us here at GPAP. So thank you very much to all of you for attending this webinar.